Thank you to BFR for inviting me to partake in this symposium. So, can everybody hear me okay? Good, excellent, thank you. I was asked to talk about classical approaches in genotoxicity assessment, and my interpretation of this is what we actually do at the moment. So this is our current approaches, and I'm going to talk mainly from the perspective of the approaches in food chemical safety assessment, because those are the areas that I know most. But of course, the basic scientific principles are relevant in other areas. Now, how do I do this? Sorry, I should have asked this first.
Yeah, you should. Go ahead with this.
This one? Okay, thank you. So this will be the focus of my presentation this morning. I will talk about the various guidance that is used, mainly in EFSA, the testing strategy that has been devised by EFSA quite some time ago now, 2011, and then since then, various issues that have arisen for which EFSA has provided clarification in its guidance, in relation to the in vivo unscheduled DNA synthesis assay, demonstration of bone marrow exposure in the micronucleus test, and annugenicity. And then most recently, we have produced a document on reliability and relevance assessment of genotoxicity data. I will then move on to the margin of exposure approach for substances that are genotoxic and carcinogenic, and just give my perspectives, take the liberty of giving my perspectives on the challenges for future developments in terms of quantitative assessment of genotoxicity data. So as I say, the main focus is on chemicals in food, but it's relevant to other areas, although obviously there are different regulatory requirements and restrictions in different sectors. So the EFSA guidance that's of relevance to our current approaches is the genotoxicity testing strategies from 2011, the clarification of some aspects of genotoxicity assessment from 2017, guidance on annugenicity assessment from 2021, and then this recent technical report on reliability and relevance. And then, as I say, I will talk about the margin of exposure approach. For WHO and JECFA perspective, our guidance Bible, we talk about it, is Environmental Health Criteria, volume 240, which was initially published in 2009, but various chapters have been updated more recently, and particularly the chapter on genotoxicity testing and the chapter on benchmark dose modeling. So those are relevant. So this is the 2011 EFSA strategy. The objective of genotoxicity testing is defined as being to identify substances that could cause heritable damage in humans, to predict potential genotoxic carcinogens where carcinogenicity data are not available, to contribute to understanding of the mechanism of action of chemical carcinogens where carcinogenicity data are available, obviously. And the importance of these different objectives vary for different sectors. Most of my experience has been with chemical contaminants, which are unavoidable and not subject to approval. So the approach you take with substances that are subject to authorization, approval for use can be different, but as I say, the basic scientific principles are the same. The testing aims to identify hazard in relation to the different genotoxicity endpoints, and Tanya has already touched on this. So these endpoints are induction of gene mutations, induction of structural chromosome aberrations, i.e. clastogenicity, and induction of numerical chromosome aberrations, i.e. annugenicity. And no single test can simultaneously provide information on all of these endpoints. So we have a basic battery, a tier 1, which in the EFSA strategy is defined as a bacterial reverse mutation test in Salmonella and E. coli, commonly known as the Ames test, and that is obviously aiming to detect gene mutation. And then we have the in vitro mammalian cell micronucleus test, which considers structural and numerical chromosome aberrations. If the outcome of those tests is negative, then under the EFSA strategy, no further testing is required unless the available information indicates the inadequacy of the in vitro test systems. If the outcome of those tests is positive, either for one or the other or both, then in vivo testing is required. And as I say again, requirements may differ in some sectors. So in some sectors, three tests are needed in this basic battery. And in some sectors, in vivo testing is required regardless of the outcome of the in vitro testing. But this is the EFSA strategy. So the follow-up of positive results has to be selected case by case, depending on, for example, the in vitro test results, which of the studies have given positive results, any information on structure-activity relationships, metabolic and toxicokinetic considerations, particularly if it's known whether metabolic activation is required for the genotoxicity to be expressed, and the potential for site of contact effects, for example, in the GI tract, if we're talking about chemicals that are present in food. For chromosome aberration, this follow-up test is recommended to be the in vivo micronucleus test in bone marrow or peripheral blood, for which obviously there is a OECD guideline. Just to follow up on gene mutation positive results, then we have the transgenic rodent gene somatic and germ cell mutation assays, and I could add pig-A to this. Apologies, I didn't update this particular slide. And then we also have the in vivo mammalian alkaline comet assay, which can be performed in a range of tissues, and that's important if you suspect site of contact effects, because it can be performed in the GI tract. Liver is going to be important if you have substances requiring metabolic activation. Other tissues might be required as considered relevant, and again we have an OECD technical guidance document for this. The outcomes of the in vivo genotoxicity testing, if they're negative, and that means probably negative, for the bone marrow micronucleus test, that means there has to be evidence of target cell exposure, then no further testing is required. And if they're positive, then we have identified a genotoxic hazard, and no further testing is going to be performed, and the action will depend on the sector, and in lots of regulatory authorities that means that approval will not be given. Substances that are positive in tests in somatic cells are assumed to reach the germ cells and to be germ cell mutagens. And in the EFSA strategy, even if there is a negative carcinogenicity study, genotoxicity in vivo in somatic cells is considered an adverse effect per se. So mutagenicity is a hazard in its own right. It's not simply a mode of action of carcinogenicity. A conclusion on genotoxic hazard indicates a health concern, and at the moment, no quantitative risk assessment is performed. To remind you of the basic risk assessment paradigm, which we're challenging at the moment, genotoxicity assessment comes under hazard identification. And if positive, then we stop there. We've identified a hazard, we don't do more. But if we have carcinogenicity data, then we will proceed to hazard characterization and do a dose-response analysis of the carcinogenicity data. and then compare that with exposure assessment in order to do risk characterization. And that I will come to towards the end of my talk. Clarification of some aspects related to the genotoxicity assessment as performed in EFSA. So, first of all, there was the unscheduled DNA synthesis assay as conducted in vivo. There used to be an OECD guideline for this, but that has been withdrawn. And the view is that UDS detects induction of unscheduled DNA synthesis in cells that are not dividing in the liver of treated adult rats. The test is designed to respond to substances that induce a type of DNA damage repaired by excision repair, but not by other mechanisms, and not unrepaired genetic damage. So it's an indirect measure of genotoxicity. Because it's quite specific in what it detects, a negative in vivo UDS alone is insufficient to rule out in vivo genotoxic potential. And the EFSA guidance states UDS has a limited use for cells other than liver, and its sensitivity is being questioned. So on that basis, EFSA concluded on the usefulness of the in vivo UDS assay. For existing UDS results, if they're positive, then that contributes to the weight of evidence. If they're negative, they're not sufficient alone, and other more reliable tests are needed in order to complete the assessment, and that would be the gene mutation assays in transgenic animals or the in vivo COMET assay. And for future work, it's recommended that UDS should no longer be performed because it's not possible to define where it would be useful and give information that's not detected by the other assays. Then we talked about verifying exposure of the bone marrow in a negative in vivo micronucleus test. The intention of this is if you have a negative result, you can't be sure that it's a true negative unless you can demonstrate that the bone marrow has been exposed. And this applies to both whether you're measuring it in the endpoint in bone marrow or in peripheral blood, because it's the bone marrow that is the target tissue. So the first evidence, the most strong evidence, is toxicity in the bone marrow in the actual micronucleus test as performed. And that's generally seen by a change, a decrease in the ratio of polychromatic to normochromatic erythrocytes. A second line is toxicity studies separate from the bone marrow study, demonstrating effects in the bone marrow. A third line is absorption, distribution, metabolism, excretion study, demonstrating that the substance and or its metabolites can reach the bone marrow. Fourth line is systemic toxicity observed in the micronucleus test itself. Then we have systemic toxicity observed in toxicity studies using the same route of administration and the same species used in the micronucleus test, but observed in other studies. Then we have substances or metabolites detected systemically in the ADME studies. And finally, substances detected systemically in an appropriate blood plasma analysis with an appropriate analytical method. And here it is somewhat challenging to define what sufficient level of exposure means. So these are all different lines of evidence that contribute to determining that the bone marrow micronucleus test has given true negative results. And then we gave some advice on establishing health-based guidance values. So this could be called reference values in some sectors, acceptable or tolerable daily intake in other sectors. And it's only considered appropriate to establish a health-based guidance value when the overall evaluation leaves no concern for genotoxicity in vivo, or the genotoxicity is due to doses resulting in saturation of detoxification pathways, or the substances interact with molecular targets other than DNA. And for those second two aspects, robust data on the underlying mode of action are essential. So then, based on the overall assessment, if concern for genotoxicity remains, derivation of a health-based guidance value is not considered appropriate. And we define this, the health-based guidance value, as an amount that can be ingested over a defined period of time without appreciable risk to health based on the available evidence. I should note here that the IPCS document, WHO document EHC 240, also specifies that derivation of health-based guidance is not considered appropriate when there's a concern for genotoxicity. Moving then to annugenicity, and this is one of our exceptions that we have that might allow a health-based guidance value to be set. So, as I've said, clastogenicity substance induce structural chromosome aberrations, and that generally happens through DNA breaks. In contrast, annugenic substances induce numerical chromosome aberrations through interactions with cellular targets other than the DNA, such as proteins involved in the segregation of chromosomes during mitosis or meiosis. I got these in the wrong way around. Yeah, so it's theoretically possible that substances that induce gene mutations or clastogenicity can interact with DNA with a linear dose-response relationship, i.e. one singular molecular interaction can induce mutations. Now, we know that that's highly unlikely in practice, but we can't define where those threshold levels would occur. For annugens, because they have non-DNA targets and induce abnormal chromosomal segregation, an interaction with a variety of molecular and structural targets is required in order to produce that abnormal chromosome segregation. And examples of this are colchicine, carbendazim, mebendazole, and nocodazole. So we have a critical number of molecular events, and very often we see a steep dose-response relationship for those, for the annugenicity endpoint. So on that basis, it's concluded that a thresholded mechanism is plausible and a health-based guidance value can be established. And that needs to take into account the entire toxicological database in order to determine whether annugenicity or some other effect, for example, neurotoxicity, might be the effect that's seen at the lowest doses, what we call the most sensitive effect. So this then is the scheme that the EFSA have come up with for annugenicity assessment. If we have positive in vitro micronucleus plus S9 and or minus S9, that could be due to clastogenicity or it could be due to annugenicity. So annugenicity mechanism needs to be confirmed by staining for the kinetochore, which reveals whether or not annugenicity is involved. If we have in vivo micronucleus in mammalian erythrocytes, if that's a positive result, then we go to risk assessment, provided there's negative results for clastogenicity and gene mutation. If we have a negative result in the in vivo micronucleus test, we need to look for evidence of bone marrow exposure, as I've already described. And if there is evidence, then it's considered to be a true negative and there's no safety concern. If there's no evidence of bone marrow exposure, then we need further testing. Now, for substances that are subject to metabolic activation in order to produce a positive result, the micronucleus test in the liver would be appropriate. Otherwise, for direct-acting substances, there's a concern about possible site of contact effects, and a micronucleus test in the GI tract might be considered appropriate. However, we recognize that we don't yet have technical guidance for these tests. The liver micronucleus test is perhaps closer to development of a technical guidance. For the GI tract, we need quite a lot more work to be done before that can be adopted as a standard method. So, anugenic compounds detected in vitro exclusively or predominantly in the presence of liver S9 suggest the involvement of liver-specific metabolites, and they can be evaluated in the liver micronucleus assay. Anugenic substances inducing increased in vitro micronuclei in the absence of S9 can be evaluated with the micronucleus assay in the GI tract. And we need more data to support development of technical guidance for these assays. But EFSA has said that even in the absence of technical guidance, they will consider such data in a weight of evidence approach. And then, most recently, as I say, we have this document on reliability and relevance. This is increasingly being applied in many areas of risk assessment, not just genotoxicity assessment. It's also mentioned in EHC 240, but with limited detail on how it should be performed. So EFSA came up with this document that aims to harmonize the approach that's taken across the different activities in EFSA. This was developed by the cross-cutting working group on genotoxicity with the aim that it's applied by the different panels and units of EFSA. So, first of all, there's the assessment of reliability, which, based on the principles of Klimisch, evaluates the inherent quality of a test report or publication relating to preferably standardized methodology and the way that the experimental procedure and the results are described to give evidence of the clarity and plausibility of the findings. And it includes assessments of whether compliance with relevant OECD test guidelines has been performed, and also whether the methodology is standardized and the completeness of the reporting. And we have these 4 criteria based on Klimisch. 1 is reliable without restriction. 2 is reliable with restrictions, i.e. with some limitations to the approach. 3 is not reliable, and 4 is not assignable because there's insufficient information. We then consider the relevance of the test system, and this is assigned as being high, limited, or low, and that takes into account the genetic endpoint, and higher relevance is given to studies providing information on apical endpoints for genotoxicity, such as gene mutations and structural and numerical chromosome alterations, rather than indicator tests. Supporting information may be obtained from indicator assays. The exception is the in vivo COMET assay, which is considered of high relevance when applied as follow-up to a positive in vitro result. And there is, as I've already said, a technical guidance there, so that ensures the quality of the test. Tests defined as being of high relevance for hazard identification, bacterial reverse mutation test, Ames test, mammalian cell mutation tests in vitro, micronucleus tests in vitro and in vivo, chromosomal aberration tests in vitro and in vivo, the COMET assay in vivo, and the in vivo mutation tests in transgenic rodents and the PIG-A assay. Tests with lesser relevance could include the COMET assay in vitro, the gamma H2AX assay, growth arrest and DNA damage assay, changes in expression and or functions of genes involved in DNA repair, unscheduled DNA synthesis, in vitro or ex vivo, recognizing the limitations that I've already mentioned, sister chromatid exchange assay and formation of DNA adducts in vitro or in vivo. But it's stressed that obviously formation of DNA adducts provides useful mechanistic information, in particular to clarify whether the genotoxicity is due to a direct DNA reactive mechanism, and thus it's considered in the weight of evidence of genotoxicity assessment. We then consider the relevance of the study results, which again are defined as high, limited, or low, and that takes into account the relevance of the results, the relevance of the test system, i.e. the two considerations that I've already mentioned, and then can take into other possible considerations, such as the route of administration. Oral is obviously considered most relevant for substances that are going to be consumed, although IV might also be relevant in some circumstances because it ensures exposure to the bone marrow. Other routes are maybe less relevant for oral exposure, but may be more relevant if you're assessing, for example, inhalation exposure. And the weight of evidence, as defined in this EFSA report, considers only the results with high or limited evidence. So the low relevance results are not taken further. Mode of action studies are considered as supporting evidence, and it's not a question of saying we have 6 positive studies and 4 negative studies, therefore it must be positive. It's a qualitative method based on expert judgment. So that was genotoxicity as we currently assess it. I'm now going to move on to what we do if we have carcinogenicity data. So this document is now nearly 20 years old. It's the EFSA approach to risk assessment of substances which are both genotoxic and carcinogenic. Nearly 20 years old, but it's still very influential, I think, and it's now well recognized that the approaches defined in this document are being used. So the margin of exposure is defined as the ratio of a defined reference point, which is also sometimes known as the point of departure, on the dose-response curve to the estimated human intake. The preferred reference point is the lower confidence interval of the benchmark dose, the BMDL, resulting in a 10% increase in tumor incidence derived from a carcinogenicity study. And now I stress the 10% increase in tumor incidence because this is quantal data. It's based on the incidence, so it's the number of animals that get tumors. And when we do benchmark dose modeling, it's quite different if we're looking at continuous data, which most genotoxicity data are continuous, so they're not comparable to carcinogenicity data. The larger the margin of exposure, the lower the concern. And EFSA considered margin of exposure of 10,000 is of low concern based on the default 100-fold factor for species differences and human variability in toxicokinetics and toxicodynamics, which is generally applied in risk assessment of chemicals. And then an additional 100-fold to allow for additional uncertainties in the carcinogenic process, and because the BMDL is not a NOEL when considering substances that are genotoxic. That's because we have to go to a 10% incidence in order to detect a significant effect, but that's based on a relatively small number of animals, maybe 50, 60 animals. You could probably detect significant increases at much lower levels if you had a much larger population of animals, but we don't do that. So we don't know what the shape of the dose-response relationship is between the doses that are actually tested, below the doses that are actually tested in the animal studies. Now, EFSA has stated that this is not a basis for allowing deliberate additions of foods or to allow foods to be used earlier in the food chain. So not as food additives, not as pesticides. But it provides advice to risk managers to help them deal with, for example, contaminants in food that are unavoidably present. And obviously, the smaller the margin of exposure above 10,000, then that provides an indication that risk management action is required, that regulation is required. I should stress that we're talking here about substances that are genotoxic and carcinogenic. By that we mean substances that are genotoxic by a DNA reactive mechanism, not all types of genotoxicity. Because as I've said, for annugenicity, we can assume a threshold mechanism. So this doesn't apply to annugens. It applies to substances that cause gene mutations and classigenicity via DNA reactivity. So, dose-response modelling. I took this picture from EHC 240 because it demonstrates that with different models, these 9 pictures are the same data set assessed by different models. And you get different results from different models. So the modelling software has now been developed to allow a modelling average approach to be taken, which means that the goodness of fit is taken into account in averaging the data in order to come up with a model average BMDL from the available data. But future issues with the margin of exposure approach could be that there is a need to provide advice on substances for which it's not possible to calculate a BMDL for carcinogenicity. This might be because there's high incidence of tumours at all doses in the cancer studies that have been performed, or it might mean there's no carcinogenicity data available. And we have to anticipate that fewer carcinogenicity studies will be performed in the future because of the expense and resource and animal welfare considerations. So we're going to be trying to use other types of data. And there's always this little niggle here, what about germ cell mutations? Does the margin of exposure for genotoxic substances adequately cover germ cell mutations? So I'm going to take the liberty of defining what I see as key issues if we move to modelling of genotoxicity data. So which data set, which endpoint is most sensitive or relevant? How do you define that? Which species? Which cell type? For an in vivo study design, do we have single dosing? Do we have repeat dosing? Are the data suitable for dose-response modelling? And if we only have in vitro data, how do we extrapolate to in vivo? For the benchmark response, which sometimes is referred to as the critical effect size, this is continuous data. This is not incidence data. And the latest EFSA guidance and also the EHC 240 guidance both refer to the BMR should reflect something that's biologically meaningful, not just statistically significant. So how do you define a relevant BMR for genotoxicity data? I know there were some proposals in the recent paper that BFR produced, which was very useful, but we need to move to consensus on how to do that. And then how do we interpret the margin of exposure that results? Can we simply extrapolate from carcinogenicity data based on statistical comparisons of BMDLs from genotoxicity data and BMDLs of carcinogenicity data? Or should we be trying to develop a scientific approach to interpreting the MOE? Because there is some scientific basis to the MOE that we use for carcinogenicity studies. So those are the issues that I think need to be addressed. So my conclusions: in vitro genotoxicity tests are currently aimed to identify genotoxic hazard. The in vivo genotoxicity tests aim to determine whether the genotoxicity hazard identified in vitro is expressed in vivo, and there's currently no quantitative dose-response assessment of the genotoxicity data. The MOE approach for substances that are genotoxic by a DNA reactive mechanism and also carcinogenic is well accepted and has been used for almost 20 years. An analogous MOE approach based on genotoxic potency would provide a valuable tool for risk assessors and regulators. But depending on the approach taken, it's possible to generate many different BMDL values for one substance, and we need a systematic and transparent approach in order to avoid ulterior motives influencing the way that the modelling is performed. So I would advocate that the latest guidance on BMD modelling, such as that from EFSA and from EHC 240, should be taken into account. And development of an approach for genotoxicity data should take into account the lessons that we've learned from MOE for carcinogenicity. And then this challenge is how to interpret the value of the MOE. So, on that note, I would like to thank EFSA for all the support, and especially colleagues on the EFSA working group on genotoxicity for their input into the work behind this presentation. Thank you.